We are discussing the serine threonine specific protein kinases. We had discussed about different classes of these protein kinases, structure of protein kinases, and then reactivity of these protein kinases. And basically, protein kinase A, protein kinase B, protein kinase C, and protein kinase G, if you remember that we had discussed in the classification of these protein kinases. So among these, protein kinase A, we had discussion in the last class. And next one is protein kinase B. So protein kinase B is also known as AKT. Okay, and this AKT is the main protein kinase that forms the signal transduction of PI3K AKT axis. So PI3K AKT pathway is a very important in the cell, which is responsible for and which is controlling cell proliferation, cell survival, and cell death pathways. Understood? So these are very important. So signal transduction in the cell is a very continuous process. That's what we are discussing. And signal transduction, biochemistry students, we are studying as far as the mechanism. And here, which protein is connecting to which protein? Which protein kinase connecting to which protein kinase? These are all proteins, these are kinases that form the basic signal transduction skeleton. Okay. So that is the knowledge that you are going to acquire in this topic. And you all should remember, practice these different names, and remember time to time, recollect how these protein kinases are linked one to one. Okay. So there are so many new names, right? Because you're all beginners and it takes time for you to practice. So you have to keep practicing. Take empty papers and rough papers and try to recollect and put it on the paper. Then only you can remember it properly. So you have to practice all these pathways and different names that you are coming across. So we are going to discuss today PI3K AKT axis mainly. Then we are going to discuss within the this presentation about the classification of PI3K and its signaling pathway, then AKT kinase. So PI3K is the initial one, the upstream signaling event, and AKT is the downstream target of this PI3K. Understood? So upstream, downstream, this terminology you, I think, getting. If you are not getting still, you have to stop me and ask me. So PI3K AKT axis pathway, mainly it is because of the AKT, which is another serine threonine protein kinase. So remember, right, serine threonine protein kinases are the first one which were identified, then tyrosine kinases, and these kinases were the first kinases which were reported and studied in detail. So serine threonine, this AKT is one of the serine threonine specific protein kinase. So AKT is nothing but PKB. PKA already we discussed. What is PKA? Another name? Can anybody tell? Protein kinase A is where is Sangeeta? Can anybody tell? Sanjana?
Yes, sir.
What is protein kinase A? Other name?
CAMP dependent protein kinase.
Very good. Okay, but when I ask in general, nobody is telling means you can tell. Okay, so that's what. So protein kinase A is CAMP dependent because its activity mainly related with the CAMP levels. Okay, so likewise now protein kinase B is nothing but AKT, and AKT is the downstream of PI3K. Downstream, upstream, all of you know. Downstream means the effector. So if you write a reaction, forward arrow, so the before the forward arrow is upstream one, effector, the substrate is the downstream. Okay. So now PI3K, PI3 kinase catalyzes the phosphorylation at 3' position of the inositol part of various phosphatidylinositol derivatives. So PI3K kinase mainly it is responsible for phosphorylation of phosphatidylinositol derivatives. The products of the reaction like phosphatidol inositol. This is the way to write for phosphatidol inositol 3,4,5 triphosphate. So phosphatidol inositol triphosphate serves as, this is phosphatidol inositol 3 kinase. This is not triphosphate. This is triphosphate. Okay. After phosphorylation by PA3 kinase, you are getting the product as phosphatidol inositol triphosphate. This serves as a membrane localized second messenger. So this is the second messenger in lipid signaling, mainly phosphatidol inositol triphosphate is a membrane localized second messenger, and this is responsible for the downstream signaling events. How it is formed? When PI3K is becoming active and phosphorylates, then it is responsible for formation of phosphatidylinositol 3,4,5 triphosphate. So this PI3K product is this one, and this PI3K is of three types. So 1, 2, and 3 types. Fundamental role of PI3K-AKT pathway, mainly as we discussed, this pathway is responsible for controlling cell proliferation, cell growth proliferation. So growth and proliferation. So proliferation means becoming multiple number of cells. That is nothing but proliferation. Growth means increase in number and increase in size. Okay. But only increase in number is called cell proliferation. Always growth refers to increase in the number of cells and increase in the size of the cell. Like growth means for individual, like human beings supposed to say, they are growing means increase in the size. Then after certain time, they will have kids also. That is increase in number, like that, simply. So that way, microbial cells also, they will increase in size and then increase in number. In our body also, tissues having the cells, after its formation, slowly it increases in size. If you recollect, we discussed that when it increases in size, these somatic cells will come in contact with the surrounding cells. And normally, when the cells are coming in contact with other cells, that is called contact inhibition. For normal cells, when the size has become enough in such a way that it is touching the surrounding cells will send the signals like you have grown enough and now you have to stop your further growth. Then aged cells, those are called aged cells, that will then undergo, because of contact inhibition, cell death by programmed cell death, apoptosis. This is the physiology. So when these signals become aberrant, means cells become like grown in the enough grown in size, but they are not going to undergo apoptosis. That is lack of apoptosis that leads to cancer. So cells are proliferating, but they are not stopping. When there are signals, contact inhibition signals, which are responsible for cells, you have enough growth, you go for division if it is active. If it is not capable, then it has to undergo for apoptosis. So cell division, that is cell proliferation, that is also through these signaling pathways. So this particular signaling is mainly controlled by this PI3K-AKT pathway. Understood? So along with this, the cell proliferation, survival, and apoptosis pathways, this also responsible for insulin-mediated effects. Insulin-mediated effects, that is glucose metabolism. Those effects also mediated by this PI3K-AKT pathway in the cells. Okay, so you have to appreciate this. Then only that depends on your passion. Okay, then you have to practice and remember this PI3K AKT pathway. So the classification of PI3K, this may be for 2 marks question you can expect. So PI3 kinase is having about 15 different kinases which can be classified into overall 3 classes. And then there is a fourth class, that is PI3K related kinases. Class 1, class 2, class 3. This is again you have to practice 1, 2, 3, 4 times to remember. Okay, depending on some people may be good in grasping. So depending on you yourself should analyze and practice. So three different classes are known along with the fourth class, that is PI3K related kinase. So fourth class of PI3K related kinases are nothing but the larger enzymes like mammalian target of rapamycin, mTOR. Okay, this is very important in biochemistry signal transduction research. If you see this, you can see mTOR. Now you know what is mTOR. mTOR is nothing but class 4 form of PI3 kinase. And also, this class 4 includes DNA repair enzymes like DNA dependent protein kinase, DNA PK, ATR, ATM. These are all DNA repair enzymes. These are nothing but class 4 PI3K related kinases. Understood? Now, class 1 PI3Ks are mainly group of kinases which are becoming active in growth factor mediated signaling. So, growth factor are the factors when they bound to the cell surface receptors, they instruct the cells. Now you have to undergo for the cell division. Okay. So that type of growth factor mediated, even in case of wound also, when wound occurs, there is lot of growth factor signaling that responsible for cells to divide and heal the wound. Okay. So those are all tyrosine kinase receptors, or it may be metotropic, that is G protein coupled receptors. These, when they active, they can mediate the activation of class 1 type of PI3K kinase effects. Understood? So this class 1 PI3K kinase are heterodimeric enzymes, so it's the catalytic and regulatory. So there are four different type of catalytic subunits and five regulatory subunits. So these four catalytic are P110, alpha, beta, gamma, and delta. Those are four different catalytic domains are there in class 1 PI3Ks. And in regulatory subunits of PI3K, we have P85, alpha beta, then P55, 84, and 101 types. So there are different subunits of four catalytic and five regulatory subunits that constitutes class 1 PI3Ks. So these class 1 PI3Ks are getting activated when tyrosine kinases or G protein coupled receptors are getting active. And this finally generates PI3K substrate is phosphatidyl inositol 3,4,5 triphosphate. Correct? PI3K results in formation of phosphatidyl inositol 3,4,5 4,5 phosphate, triphosphate, which is membrane bound molecule. So this in turn formation results in the activation of AKT now, AKT pathway downstream signaling and Rho family GTPases. Understood all of you? So this is the way class 1. Now next remaining is class 2 and class 3. These are all responsible for intracellular trafficking through the same phosphatidyl inositol 3 phosphate and 3,4 diphosphate. Class 1 is mediated by triphosphate, whereas class 2, class 3 mediated by 3 phosphate and 3,4 phosphate. So for the next discussion of PI3K AKT pathway, only we are considering class 1 PI3K mediated effects because they are the one which are responsible for important cell, physiological event, that is cell proliferation, cessation of apoptosis. Understood? Okay, if you understood, then we will move on with the pathway as we discussed. So different classes of PI3K responsible for class 1 type of PI3Ks are activated by the either receptors. So these are hormones or growth factors which are binding. Even hormone itself can be a growth factor. Growth factors and hormones, nothing but they can be called as mitogens. So that's why it is called mitogen signal transduction. RAS. Okay. So here you have growth factor mediated. These are all receptor tyrosine kinases. This already we discussed. Receptor with intrinsic tyrosine kinase activity. Then another one is G protein coupled receptor. This is metabotropic receptor, serpentine receptors. You are well aware about this. So there are which stimuli also you are aware. Now what happens? These are the transmembrane receptors which can activate PI3K. So three different ways it can. either through transmembrane receptors of growth factor receptors or beta gamma dimer of G protein coupled receptor signaling pathway mediated or activated molecule beta gamma dimer can activate, or the RAS pathway, that is mitogen activated protein kinase, MAP kinase pathway, RAS. This can also act and activate PI3K. So this activated PI3K is responsible for formation of phosphatidol inositol 3,4,5 triphosphate. This is formed from diphosphate. So diphosphate is phosphorylated by this PI3 kinase to triphosphate. And this triphosphate, so this signaling negatively regulated by this P10. How? This P10 means phosphatase tensine homolog. This is one of the protein. So all these things you have to remember, recollect as I said, okay? These are all new terms for you. So this P10 is responsible for hydrolysis of this phosphate, reversal, so that there is no PI3K-AKT pathway. Understood? So this P10 is responsible for reversal of this phosphatidol inositol triphosphate to diphosphate. So if diphosphate means there is no signaling, but triphosphate formation in the membrane, this is associated with the membrane, and this is identified by the through pH domain of the different signaling proteins, including AKT protein. Understood all of it? AKT next we are going to discuss. If you get the question for only PI3K activated pathway, this is the one you have to write. So these different signaling proteins, which are the downstream of activated phosphatidol inositol triphosphate, is then responsible and controls cellular proliferation, cell growth, and anti-apoptosis. Understood all of you? So this way, this particular pathway is very important in case of cancers. So what is happening in cancer? These are proteins. So proteins are formed that from the transcription translation, that is from the DNA. Now when DNA undergoes mutation, if there are any functional gain in mutation, these are called functional gain in mutation or functional loss of mutation. So when they gain mutations that are responsible for oncogenes, those are called oncogenes. So oncogenes will be having role in this functional gain in mutation. They are nothing but the participants of this signal transduction pathway. So what happens? Their protein expression will be constitutive, so that continuously that protein pathway will be active. So cell proliferation continues. It should stop. So this negative signals will not be working. Suppose here phosphoten this protein. Now here this protein is a checkpoint protein. Okay, enough. Like when you are filling your bucket, okay, for example, at your home, you are working right in the kitchen, you are helping your mom at home. So when you are filling some bucket or something, when it filled, then you have to stop it. That is a negative regulation. You need to fill, you need to stop also. So that stop signals when they fail also is pathology. That is nothing but cancer. So now these stop signals, here, these are called tumor suppressors. They will be checkpoint proteins. They will check the cell whether it is in a normal pathway or is it aberrant. So this should also be functional properly. These are called tumor suppressors. And these constitutively, if the mutation is responsible for expression and mediated signaling pathway, those are called oncogenes. Understood? So cancer is nothing but because of the oncogenes, oncogenic expression, and loss of tumor suppressor functions. Understood? So this way now, this PI3K-AKT pathway is having very important role in all the cellular physiological effects, and anything abnormal means it leads to aberration. So this is what now PI3K pathway. See here how signal transduction very difficult. No, there are so many number of proteins which are interconnected. Now, as a biochemistry student, for drug discovery program, what we do? See here now, this AKT is the final molecule which is being regulated or controlled by number of proteins. So your drug targeting this AKT can be very effectively that it can control, because it is a central molecule which is regulating many number of events. So our pathway we are going to discuss here. See here PI3K. This phosphatidylal inositol 3 kinase which we discussed, PI3K, the upstream signaling protein is binding to the activated receptor after it is has become phosphorylated. So phosphorylated receptor is identified by this PI3K and responsible for, as we discussed, PI3K is responsible for formation of phosphatidylal inositol 3,4,5 triphosphate. This is the product, and this is again responsible for the binding of PDK1 one protein. Next we'll be seeing this PDK1, and this responsible for activation of AKT. Understood all of you? So this way our PI3K AKT signal. So PI3K is activated by three. This is the explanation for earlier discussion. So we have seen that activation by transmembrane receptors, activation in RAS pathway, then activation by G beta gamma dimer. The PI3K pathway can be activated by growth factor receptors either by direct interaction via adapters like insulin receptor substrate. As we discussed, even in insulin metabolism also, this PI3K is very important. So these all can activate these growth factor receptors, and also through these growth factor receptors are in turn responsible for activation of RAS mediated pathway. So through this RAS pathway also responsible for controlling this PI3K. Now another route is G beta gamma dimer, that is G protein coupled receptors. When they become activated through G beta gamma dimers also, they can activate this PI3K signaling. So the product of PI3K, as we discussed, it is formation of phosphatidylal inositol 3,4,5 triphosphate, and this product is recognized by the pH domain of various proteins. Understood all of you? So overall, the activation of PI3K stimulates cell growth proliferation and inhibition of apoptosis, as we discussed. So this is mainly suppressed by PTEM. So this is all the explanation you have to write for PI3K pathway if it is asked. Next one is AKT. AKT, if you see, that is the downstream of PI3K. It has got three different domains: regulatory domain, kinase domain, and this is plexstring homolog domain, homology domain. So this is identified by this your substrate form from PI3K, phosphatidyl inositol triphosphate, will bind to through pH domain. This is pH domain containing protein is AKT. So earlier we have discussed that through pH domain containing proteins, several proteins of pH domain containing proteins can interact with this phosphatidyl inositol triphosphate, right? So this recognize pH domain of the AKT kinase. Then PDK1, remember PDK1 also is there. That is also responsible for phosphorylation of this kinase domain, and mTOR is also responsible for phosphorylation of regulatory domain. So phosphorylation by PDK in kinase domain and phosphorylation in regulatory domain by mTOR is responsible for full-fledged activation of ATK kinase, and this is negative regulation, PPTA. So the major downstream effect of this PI3K kinase is AKT. That's how it is PI3K AKT pathway. So AKT now, which is nothing but our protein kinase B. So AKT exists in three different isoforms, and it is contained in pH domain. So N-terminus, this is N-terminal, this is C-terminal. In case of DNA, you write 5', 3'. In case of protein, it is N-terminal to C-terminal. So pH domain, as we discussed, kinase is activated by the phosphorylation of by the PDK1, and regulatory domain phosphorylation site is phosphorylated by mTOR, and that makes the protein completely active. This is the AKT kinase domain structure if you get for 2 marks question. Easy, right? So now next is AKT kinase signaling. So any questions you have? All of you followed? Shall I stop here or shall I continue AKT kinase signaling? If you are following, then I will continue. Please respond immediately.
Followed, sir.
Okay,
Sir,
good. So AKT kinase signaling. AKT is the kinase now, as we discussed, it is the downstream of PI3 kinase. And this AKT kinase signaling pathway is responsible for the same thing, cell proliferation, anti-apoptosis, and which is deregulated in many cancers, cessation of apoptosis. So AKT downstream, once AKT is phosphorylated, it is responsible for different proteins. Again, we'll come across. So mainly different transcription factors like GREB, different protein kinase, IKB. This is inhibitor of kappa B. NF-kappa B is one important protein in the cell, and the part of NF-kappa B is inhibitory kappa B. So inhibitory kappa B, if it is active, NF-kappa B will be phosphorylation of inhibitor of kappa B is responsible for activation of NF-kappa B. Okay, so protein kinases will be like IKB getting activated, phosphorylated by AKT, transcription factor like CREB, and pro-apoptotic proteins like BAD and others like MDM2. This is a cell cycle protein. So these are the proteins which are downstream of AKT. What is upstream of AKT? We discussed. Immediately, please. Somebody should tell. What is upstream of AKT? Somebody should tell immediately what is the upstream of AKT. Sanjana?
PI3K, sir.
Yes. Okay, you can tell. Okay, I don't know others whether they are not following what is upstream and downstream. If I am asking, pointing out one student, then you people are telling. Punyashri,
sir.
you have followed or not?
Yes, sir. I'm following.
Okay. So you need to ask, otherwise it's very difficult, as I said. Through online class, it is a very difficult thing. So whether you are following or not is going to be tough. But upstream, downstream is a very simple thing now. So these are the steps of AKT kinase signaling. What is happening AKT kinase now? AKT kinase means downstream of PI3K. So stimulation of class 1 PI3Ks, that is nothing but growth factor mediated signaling. So receptor tyrosine kinases, these are the receptors with intrinsic kinase activity. So these receptors, when growth factor is binding to these receptors, this undergoes autophosphorylation, correct? This pathway already you know. So the tyrosine residues undergoes phosphorylation. Now, these phosphotyrosine residues, they serve as the attachment sites for the regulatory subunit, that is P85. So this P85 already we discussed, this is the regulatory subunit part of the PI3K. So this is the first thing, PI3K will identify through P85 and they can bind. Now, this PI3K getting activated PI3K is converting and results in the formation of phosphatidyl inositol triphosphate. This also we discussed. And this generated by PI3K, that is actually membrane bound. And through pH domain containing proteins like PDK1 and AKT, they are translocating to membrane, and they will identify this molecule. Okay, so now this is accompanied by the release of autoinhibition. When PDK1 AKT getting phosphorylated, that is responsible for release of autoinhibition. So full activation of AKT. After this, AKT is not completely active, but this complete activation of AKT is because of the phosphorylation by mTORC2. Okay, so now it is fully active. Activated AKT then phosphorylates variety of targets, as we discussed. So this is what we discussed. This is your receptor tyrosine kinase. There is a mitogen or growth factor bound, and it is identified by PI3 kinase through its regulatory subunit P85. This is responsible for converting PIP2 to PIP3 phosphate. This is the product PIP3 triphosphate, phosphoinositol triphosphate. and P10 is the negative regulation. It converts PIP3 to PIP2, so there is no PIP3 signal. But here, in the absence of P10, this is formation of PIP3. See here, these are all membrane bound. Now, this is identified by the pH plectrin homology containing domain containing proteins like PDK1 and AKT will identify these proteins. So this results in phosphorylation of AKT, PDK1, AKT is getting phosphorylated, and this phosphorylated AKT is further phosphorylated by mTOR and responsible for C2 phosphorylation, makes this protein fully active and responsible for all this. I'm stopping here. I will continue in the next class. Any questions you have, you can ask. But a very sad thing that many people are leaving in between. Earlier it was 11, now it is 9. It's not acceptable. Why they are leaving, I don't know. So I am taking attendance only at the end. So those who left, it will be problem for them. Okay, any questions? You can make it out from your side. You can write it down. Next class, you can discuss. See you tomorrow then.